Good afternoon, colleagues, and thank you for joining us for one of our Sydney ID Distinguished Lectures. We are very fortunate today to have Professor Thijs Kuiken, who's at the Department of Virus Science at Erasmus MC in Rotterdam. And I think it's fair for me to say that he's widely regarded as the world expert on H5N1. Before we start, if we could just acknowledge the traditional owners of the various lands on which we meet today and pay our respects to their elders, past and present. For thousands of years, they have cared for country. I think the multiple crises we face today reminds us all to be more aware of our ecological footprint. And I think Thijs's talk will convince you that this is also highly relevant to those of us working in infectious diseases. Thijs, without any further ado, I'm going to hand over to you. We want to have as much time as possible to hear from you and the global overview that you will provide. Just to mention to people that if they have any questions, there will be time for questions at the end, but we will have to wrap up by 5 p.m. at the latest. But if you have any questions, if you could just pop that in the Q&A, and we'll try and get through as many as possible. Over to you, Thijs.
So thanks very much, Ben. I'm very happy to be able to give this talk. In the next 40 minutes, I'd like to talk about the global expansion of highly pathogenic avian influenza virus of the subtype H5N1. And I'll give you a background to the increased risk of HPAI outbreaks in recent decades. I'll specifically focus on the global spread of one particular virus, the H5 of the Goose Guangdong lineage from 1996 to now. I'll emphasize a recent event, the incursion of the virus into the dairy cow population in the United States. I'll talk about the massive impact that this virus has had so far on South American wildlife and potentially also will have on Antarctic wildlife. I'll talk to you also about the possibility that this virus may spread to Australia and the rest of Oceania, and then briefly discuss options for response. So HPAI virus is not a naturally occurring virus in nature, but it's a mutant of low pathogenic AI virus, and the conversion from low to high pathogenic occurs in poultry. And basically the difference between these two variants of AI virus are that the hemagglutinin of the low pathogenicity avian influenza virus can only be activated by host enzymes in the respiratory and the digestive tracts of birds, and therefore the virus infection is limited to those tracts, whereas the high path version has a hemagglutinin that can be cleaved by host enzymes throughout the body, thereby allowing the virus to replicate in multiple organs and cause high mortality. So that's the basic mechanism for this change. So Murad Dingra, currently working at the Food and Agricultural Organization, made an interesting review with her colleagues about the conversion events from this low to high pathogenicity avian influenza from 1959 to 2015. And I just want to highlight three main findings that they made. And in this map, you can see the locations of the conversion events from low to high path AI in different colors in three time areas before 1996, from 1996 to 2005, and from 2006 to 2015. And the three results are that, in the first place, most of the conversion events take place in intensive poultry systems, and it's considered that there's a selective advantage within these large high density barns for the high path version to be advantageous above the low path version. In time, we see that in the second half of the period studied, there were about 3 times as many conversion events as in the first half. And this is probably related to the rapid increase in poultry production worldwide. And if we look across the globe, we can see that in every continent where poultry are kept, there have been these conversion events from low to high path, including in Australia, where there have been 7 recorded in this period studied. And I understand that today there has been a new high path AI event recorded in Victoria, H5N1. So in that sense, this talk is very timely. So this figure emphasizes the positive correlation between the number of high path AI outbreaks and the growth of the global poultry sector, measured in poultry meat production. So the blue bars indicate the number of high path AI outbreaks per decade, which are shown to be increasing at the same time as the poultry meat production per year, shown as the blue line with the blue dots, also is increasing. And at this point, I'd like to make a side track and not only focus on high path AI and really have this narrow view, but also to look at this problem in the light of planetary boundaries. I hope you are aware of planetary boundaries, and if not, this paper from Richardson et al. Showing that we are currently exceeding 6 of the 9 planetary boundaries is a useful publication to look at. And for this talk, I'll be concentrating on climate change, biodiversity loss, and land use change as planetary boundaries in which livestock production, including poultry production, are involved. This figure shows the increase in global poultry production from 1961 till 2022. And you can see that in the last 20 years, there's been a massive growth in the poultry population from 17 billion to 28 billion poultry birds at any one time. That means an annual growth of 500 million per year. The resulting meat and egg production in 1 year, 2022, is associated with CO2 emissions of 1.1 gigaton CO2 and associated with land use for production of feed for poultry of 2.3 million square kilometers. So you can see that this poultry production that has grown so big really has a massive influence on these planetary boundaries, which raises the question whether there should be a cap on global poultry production, and if so, at which level this cap should be. Associated with this massive increase in poultry production, there's also been an increase in poultry meat consumption, as shown in this graph, together with meat consumption from other livestock, pigs, beef, sheep, goats, and other meats. Part of this increase in poultry meat consumption is due to advertising. And as the well-known professor of animal husbandry from the 1980s stated, aggressive marketing methods have increased the per capita consumption of poultry meat more than 5-fold in the 3 decades after 1950, and further increase may be expected in the future. So although this poultry meat consumption worldwide has grown, there is not an equitable sharing of this per capita meat consumption. And for this talk, I've contrasted the consumption of meat in Australia, where the total consumption per average Australian is 110 kilograms, compared to the average consumption of an African, which is on average 18 kilograms per year. So if we compare that to the International Eat Lancet dietary guidelines, in Africa, the average person nearly reaches the yearly guidelines, 21 kilograms per year, whereas the average Australian consumes about 5 times more than the maximum considered meat. For Australia, I could not find what the dietary guidelines were for meat in general. However, for red meat, it was advised not to eat more than 600 to 700 grams per week, which comes to 31 to 36 kilograms per year, whereas currently the average Australian eats about 60 kilograms of red meat per year. This inequity in consumption of animal meat also correlates with big differences in the health impacts from malnutrition in different parts of the world. And one of the parameters is shown here, the protein energy malnutrition mortality rates in children, showing that there's a big difference, for example, between the East Asia and Pacific region, which includes Australia, where there are 2 children per 2,000 who die from protein energy malnutrition, compared to sub-Saharan Africa, where 43 children die from this problem. And as a response option to solve this, Marius Gilbert and others have suggested, with regards to livestock production, including poultry production, that there needs to be actions on both ends of the consumption and production spectrum. So on the end of high income countries, where there's more consumption than necessary of meat, they recommend so-called de-industrialization, so decrease in the intensity of the animal production, whereas on the side of the low-income countries, where there is malnutrition, partly due to a lack of sufficient protein, they recommend a sustainable intensification. So in this way, it would be possible to have a more equitable distribution of consumption and production without necessarily increasing the global production of livestock, including of poultry. So now I go back to specifically this HPAI outbreak of the H5 subtype. So this one outbreak, starting in 1996, has had a bigger burden of disease than all of the other HPAI outbreaks combined. So it really is a gigantic outbreak. So just to give you some figures, to date, nearly 400 million lives of poultry have been lost as a result of the infection or of culling. Tens of millions of wild birds of nearly 400 species have died from this virus. We don't know the exact numbers because surveillance for wild birds is inadequate. Similarly, probably millions of wild and captive mammals of about 50 species have died from the virus. And as for humans, since the beginning of the outbreak, there have been nearly 1,000 cases of infection recorded, of which just over 50% of the people have died from the disease. And I'd like to take you around the world from the beginning of this outbreak to the current state. So the virus infection was first detected on a commercial goose farm in Guangdong province in southeast China, where there was a 40% morbidity in the geese. And this virus the Goose Guangdong 1996 virus gives the lineage of viruses that are causing this outbreak its name, the Goose Guangdong lineage. So all the viruses that have evolved from since then come from this first isolate. So 1 year after it was first detected, this virus caused an outbreak in Hong Kong in 1997. And during this outbreak in chickens in Hong Kong, 18 people were hospitalized because of H5N1 virus infection, and 1/3 of those people died. And this outbreak was aggressively dealt with by killing all the chickens and other poultry on Hong Kong. And indeed, the outbreak then stopped. And people at that time were very surprised because the H5 subtype had never been found in people before. It was not usual for the virus, an avian influenza virus, to be directly transmitted from chickens to people. And it was also not known that it caused such severe disease and even death in people. We were able to show at that time that although the upper respiratory tract of people did not have receptors for avian influenza virus, the lower respiratory tract did, shown in the left panel here, where the brown staining shows the attachment of avian influenza virus H5N1 to the pulmonary alveolar epithelial cells. And that's why it mainly caused a severe pneumonia, as shown in the middle panel by this X-ray showing the density of the lung photo. And it was associated with severe inflammation and necrosis, as shown in the right panel, which shows a microscopic picture of a pulmonary alveolus completely filled with red blood cells, fibrin, and inflammatory cells, making breathing impossible. At the same time as the virus infected people, the virus also was found to infect cats that came into contact with chickens. And to our surprise, the virus didn't cause just respiratory disease, but it spread to multiple organs in the cat's body, shown here, and caused, as shown in the upper row of pictures, inflammation and severe necrosis in these tissues. And this inflammation and necrosis was associated with the presence of the virus, as shown in the lower row, where all the brown staining is the presence of influenza virus antigen in the affected cells, particularly in their nuclei. So not just a respiratory disease, but actually a systemic disease in affected cats. And the same thing was seen in multiple other mammalian species in the years that followed. So we take a big jump from 1997 to 2005. Until 2005, the disease was restricted to Southeast Asia, but in 2005, suddenly the virus was found in Europe, in the Middle East, and in Africa. And in Europe, the main wild bird species that were affected were water birds, as well as raptors. And in some countries, the virus was found in wild birds in absence of affected poultry. So the question was, how could this virus be carried by wild birds over such a large distance? Because the idea was that sick birds don't fly. However, although a part of the wild birds did become ill and die, another part were able to be infected without showing clinical signs, and indeed were able to fly long distances. And indeed, that was what we found to be the model for the long distance spread of the virus from Asia. It didn't go in one step from Asia to Europe. So although there are rare birds that do fly directly from Asia to Europe, that's not the usual case. Instead, the pattern that we found was that wild birds wintering in Southeast Asia would, during spring migration, carry the virus to the common breeding areas of Eurasia, namely in northern Russia, Siberia, where they mingled with birds that had arrived from Europe and infected them. And a few months later, during autumn migration, the European birds were able to take the virus back to Europe and cause infections in other birds there, including in poultry. So in this two-step cycle, the virus could be transported over 1,000 kilometers between different flyways of wild birds. The next big change that happened with this virus was that since 2021, the virus adapted to persistent circulation in wild birds. So in the top left panel, you can see the traditional epidemiology of avian influenza, where there is circulation endemically of low pathogenic avian influenza viruses in wild birds. Occasionally, these low path viruses transmit to poultry, and sporadically those become high path and cause outbreaks in poultry, and it stops there. What we see now with this Goose Guangdong lineage of the H5 virus is that in poultry, there is, in some countries, endemic circulation of high path avian influenza viruses. These viruses are able to spill over into wild birds. And now the wild birds also have endemic circulation of high path avian influenza viruses with occasional spill back to poultry. So this is a completely different situation, which means that in many parts of the world, the virus is becoming endemic in the wild bird populations. And support for this endemicity of the virus in wild birds comes from this phylogenetic analysis that was carried out by Ann Pohlmann and her colleagues at the Friedrich Loeffler Institute. And in this phylogenetic tree, you can see that the viruses that were found between 2020 in the fourth quarter down to the fourth quarter of 2021 are all closely related to each other in this B1 branch. Furthermore, you can see that there's a stepwise branching pattern during this evolution of the virus, and that there's a consecutive temporal order of the viruses starting from the fourth quarter of 2020 until the fourth quarter of 2021, indicating that indeed this virus is circulating endemically in wild birds from which these viruses have been isolated. And the particular branch of this Goose Guangdong phylogenetic tree that has become endemic is called the clade 2344b branch. So a difficult name, but one which you will come across more and more often in the rest of the talk, and also if you read about this virus as it is currently spreading worldwide. So at the same time as the virus became endemic in wild birds, it also spilled over from Eurasia to North America in 2021. And the first place where it was found was at the very eastern tip of the North American continent, at the very eastern tip of the island of St. John's, or of the island of Newfoundland in the city of St. John's, where an exhibition farm was found positive for the virus. And a few weeks before, retrospectively, a wild bird, a great black-backed gull, that had been found ill also was diagnosed with the same virus. And by phylogenetic analysis of the viruses from Europe, shown here in violet, and the viruses from this exhibition farm on Newfoundland, shown here in orange, were shown to be very closely related to each other, indicating that indeed the virus on Newfoundland came from Europe. And we hypothesize that in the same way as the virus was spreading from Asia to Europe, the virus also spread from Europe to North America, being carried in the first place from Europe across the North Sea to Iceland, Newfoundland, and the Canadian Arctic in spring to breeding areas, and in the subsequent autumn migration from those areas down to Newfoundland and other parts of North America. And indeed, the virus did continue its spread through North America until at least 2024. Already in the first few months after its first detection in Newfoundland, the virus had spread right across the continent of North America, Canada, and the United States, being found both in wild birds, shown here as green dots, as well as in commercial poultry, shown in red, and backyard poultry, shown in yellow. And this is just to remind you that not only wild birds were being infected in this period, but also wild mammals that came into contact with wild birds that were infected, mostly due to predation. So here you can see some of the total 50 species of wild mammals that have been found infected with this virus, often with fatal consequences due to neurological disease. But what we hadn't expected is that at the end of March of this year, the virus was detected in dairy cows in Texas. These dairy cows in southern United States were suffering from a mystery disease, and after a few weeks of testing, they were found to be positive for HPAI. In the first instance, there were 2 farms in Texas and 2 in Kansas, in which about 10 cows per farm were affected, indicating that the virus was also spreading from cow to cow. And surprisingly, the highest levels were not found in oropharyngeal swabs of these cows or in nasal swabs of these cows, because the CT values were very high, indicating low virus concentration. But very high titers were found in milk samples with CT levels between 10 and 20, indicating very high virus concentrations in the milk. So further research has shown that indeed the mammary gland tissue of these dairy cows is positive for this virus. And here you can see on the left an immunohistochemical stain of the mammary gland showing positive alveolar mammary gland cells positive for H5N1 virus. So the same cell types that are actually producing milk also produce vast amounts of virus. And on some farms, also domestic cats that had drunk raw milk from these cows became infected and died. On one farm, more than 50% of the 24 or more cats that were on the farm died as a result of this virus infection. So highly fatal to cats that came into contact with raw milk from these infected cows. And there's been one confirmed case in a dairy farm worker who was working on a presumably infected farm. The person did not suffer severe disease, but only a conjunctivitis, which is quite a normal situation, especially also with the HPAI viruses of the H7 subtype, which I understand is now in Australia. For people to have conjunctivitis, this person recovered after a few days, and there was no noticed spread elsewhere in the household contacts of this person. Although this is the only one that is known to be infected, it's suspected that there may be dozens of people who have been in contact with the virus through contact with cows and become infected. But this is only anecdotal information. So the spread of the virus continues to date. As of yesterday, there were 51 farms positive in the United States. mostly in Michigan and in Texas, but also in several other farms in the United States, as a result of infected cattle not showing clinical signs having been transported to other states and there infecting other cows on the farm in which they were imported. So the concern about this particular outbreak is that the virus becomes endemic in the dairy cow population, and also that it adapts better to cow transmission, and thereby makes it easier for the virus to adapt to human transmission and could start a new pandemic. However, it's also possible that this outbreak in dairy cattle dies out. Let us hope that that is the case. So from North America, we go to South America, where in 2023, the virus affected both poultry and wildlife. And here I'll concentrate on the impact on wildlife. In just 1 year, over 600,000 wild birds were found dead in association with this outbreak, mainly on the west coast of Peru and Chile, involving more than 80 different species. Besides wild birds, the virus also caused mortality in wild mammals, and over 50,000 individuals were found dead during the outbreak. And although there were 2 species, South American sea lions and southern elephant seals, that were mainly affected, more than 11 mammalian species in South America were involved. So the main carcass counts were of guanay and other cormorants, and of Peruvian boobies, as shown here. But several other species also showed quite high mortality, sometimes even important at a species level. And the highest mortality in wild mammals was seen in southern elephant seals on the east coast of Argentina, on Peninsula Valdez, where there is a large colony with over 55% of the worldwide pup production of elephant seals being produced there. And in November of 2023, on an area where many of the seals were pupping, nearly 1,900 pups were found dead, indicating that there was a higher than 96% mortality of elephant seal pups. In comparison, the year before, there had only been 0.8 mortality of these pups. And extrapolating this to the total breeding colony on Peninsula Valdez, it meant then that more than 17,000 elephant seal pups had died that year, nearly the whole breeding pup production of that year. That's really an astounding mortality rate for a wild animal population from an infectious disease. So from there, we go from South America to Antarctica. And in February of this year, the virus was found for the first time on the Antarctic Peninsula in skuas. There are 2 species of skuas in Antarctica, the brown skua and the south polar skua, both with rather small populations. And the birds that were found were both on the west side of the Antarctic Peninsula at Primavera Cape, and on the east side on James Ross Island. Partly as a result of this detection, there was an international group of scientists that went to Antarctica in March 2024 to assess the spread and impact of this virus on wildlife. So this expedition was led by Megan Dewar from the Federal University of Australia, with scientists from Europe and from South and North America. And this is the boat that they went on, a 23-meter long motor sailor that was able to go through pack ice. Here you can see as red dots the places where they visited during this 2-week period. And on one of the locations on Beak Island, they found an unusual mortality event in skuas as a result of HPAI H5. So this is the first mortality event that has been recorded in Antarctica from this virus. So out of 130 birds in total, there were 50 that were dead, and all 10 birds that were tested by onboard testing were found positive for the virus. And here you can see the triage of the carcasses that had been found on Beak Island to determine which ones were going to be examined in detail. And the birds are have a position where they have their wings spread and their head held back, which is typical for neurologic disease from HPAI. So there is concern about this virus spreading through the mainland of Antarctica and impacting wildlife. And most, if not all, of the species that are living in Antarctica are likely susceptible to disease from this virus. Yeah. So the next question is it possible that Australia and other parts of Oceania will now be infected with this virus? So the answer is yes, it is possible. So here you see the geographical distribution of seabirds and pinnipeds around Antarctica, and you can see that they form a continual band around the continent, which means that probably the virus can also spread from population to population around the continent of Antarctica. And if the virus arrives in Antarctica south of Australia, there are multiple seabird and sea mammal species that range between the islands to the south of Australia and New Zealand and the Antarctic coast to feed and to breed. And thereby, it's possible that they could take the virus with them and cause infection in Australia and elsewhere in Oceania. So I end with a number of options for response, and I could speak half an hour on this, but I'll be very brief. So basically, the emergence of HPAI viruses is linked to the intensification of the poultry sector worldwide and has major potential health impacts for not only poultry, but also wildlife and also humans. In the short term, response options are to improve biosafety and prevention practices. And if the virus becomes endemic in a region, to use vaccination rather than stamping out, which is currently the custom. For equitable long-term options for response, deindustrialization of poultry production in high-income countries and concomitant changes in dietary patterns are advised. And on the other hand, sustainable intensification of poultry production in low-income countries. In this way, to have societal benefits of poultry production that are optimized while reducing the negative impacts of the poultry production on the environment and on health. So I'd like to thank a number of people who have collaborated with me and discussed with me many times in the EFSA Avian Influenza Working Group, in the OFLU Wildlife Group, and most recently in the Australis Expedition Team. Furthermore, I'd like to thank Horizon 2020, the European Programme for Research and Innovation that have funded both VEO, which is a program for monitoring and surveillance of infectious diseases, and CAPAFLU, which is a program to study HPAI in Europe and worldwide. Thank you for your attention.
Thank you, Thijs. That was a really informative overview and clearly a major signal of concern about how we manage biodiversity on this planet. Maybe I can just start by asking whether the cow infection in the US is the only documented event of sustained mammal-to-mammal transmission. You showed us the elephant seals as well. Was the impression that there was also seal-to-seal transmission in that case? And has other sustained transmission chains in mammal species been documented?
There have been several events where there has been suspicion that there has been mammal-to-mammal transmission. So in the wild, it's very difficult to prove this because you can't rule out that there's also a high environmental burden of disease, so that each individual animal can become infected from the environment. So in the elephant seals, they were elephant seal pups that were basically just lying around, except when they were drinking from their mother. So it's thought that indeed there was pup-to-pup transmission of the virus. Similarly, in South American sea lions on the coast of Peru and Chile, there were hundreds of animals that died in a very short period of time. And again, because many were dying in groups together, it was thought that there was a mammal-to-mammal spread. Similarly, on the northeast coast of the United States, in harbor seals and in gray seals, there was a similar event where hundreds of animals were found dead with this virus, and it was considered to be mammal-to-mammal spread. All those cases, it's very difficult to say because we don't have much epidemiological information about the outbreaks. More convincing is an outbreak that occurred in Spain in farmed mink. So in Spain, there was an outbreak where a farm with 50,000 mink became infected with the virus after having contracted it from wild birds that were probably feeding on the same feed as the mink were feeding on. And there, the pattern of spread through the farm strongly suggested mink-to-mink spread. As well, in Finland, there have been massive outbreaks in fur animals, American mink, foxes, raccoon dogs, and again there mammal-to-mammal spread is likely. So yes, there have been other outbreaks where there has been mammal-to-mammal spread. In all those outbreaks, luckily, the virus has not spread to humans and caused human-to-human spread.
Thijs, I'm not sure if you know this from maybe any of the mammal autopsy studies, but there is a question whether you documented very clearly that CNS disease is often contributing to the fatality in birds, but in mammals, has CNS disease been recorded as well, or is this mostly death due to pulmonary infection? And it seems as if in the cows, it's not really a cause of disease, but they just create a reservoir. But what's your knowledge of disease patterns in mammals?
Yeah, so in wild mammals, actually, neurological disease is often the primary clinical sign that suggests to people that the animal has a problem. And in the first cases, in the differential diagnosis, rabies was considered to be the first on the list. And then when they were tested negative for rabies, they were subsequently tested for other infections such as HPAI, and it was found to be HPAI. So yes, it occurs regularly. In some cases, actually, there are postmortems carried out, for example, on seals, whereby there is no respiratory disease and the virus is not found in the respiratory tract, but it is found in the brain, causing severe encephalitis. And we know that the virus can reach the brain, both systemically via the blood circulation, but also via the cranial nerves, primarily via the olfactory nerve. And we have shown in experimental infections is that there can be direct spread of the virus from the olfactory mucosa in the nasal cavity via the olfactory nerve straight through the cribriform plate into the brain. With regards to the cows, it's a very different situation. So extensive post-mortem examinations have not been done yet, but my impression is that this is a localized mammary gland infection that remains mainly localized there, resulting in a decrease in milk production, but not in general, not in severe disease of the dairy cows. So here that's a very different situation.
No, thank you. That's informative to be aware of the diversity of disease. And there was a comment that maybe that also explains to some degree why the pups, the elephant seal pups, if that was milk, if the mothers could have given it to the pups through milk, if that could have contributed to pup deaths. Not at all.
Yeah, it's possible. Actually, there has been an experimental study carried out with human influenza H5N1, the cause of the pandemic virus in 2009, whereby ferret pups were experimentally infected with this virus and then allowed to suckle. And they then transmitted the virus to their mothers, which subsequently developed a mastitis.
Interesting.
And this study was carried out as a result of the observation that some women during the 2009 pandemic developed mastitis.
We've commented in the beginning that there's been a detection of H5N1, which seems also a highly pathogenic strain in an egg farm in Victoria. But there's also been a notice that we've just had our first H5N1 case in a human returning from overseas visit, a child, I believe. My impression from what you've said is that no, humans are unlikely to introduce the infection into Australia. It's really the wild bird populations which is most likely to cause widespread. But it is fascinating that we've been spared the introduction to until now, because there's migratory bird routes all the way from Siberia down to New Zealand and Australian coasts. Has the impression been that those birds, the wading birds, haven't been as badly affected? And if we're going to get infection, that it'll come from the south rather than from the north? That was the impression from your presentation.
Yes. It seems as if water birds, particularly dabbling ducks, perhaps swans, perhaps geese, are the ones that are really mainly involved in the long distance spread of the virus in Asia and in Europe. And wading birds, shorebirds, can be infected, but don't seem to be so much involved in the spread of the virus, although there may be exceptions. So one explanation may be that although there is extensive migration between Australia and Asia and New Zealand of wading birds, that is not so much the case for ducks, geese, and swans. So in this sense, it may be that Australia and other parts of Oceania have been spared. The situation is now different because seabirds can definitely also carry the virus over longer distances. And the suggestion is from the finding in skuas is that skuas are involved in the spread of the virus in Antarctica. And skuas and other seabirds do move between Antarctica and the islands south of Australia and New Zealand. And in this way, it is possible that they could spread the virus from the south. So it's coming in through the back door.
Yeah. There's also been questions around how best to prepare. You mentioned vaccines more for its poultry application, but are you aware of any human vaccines that's ready or in preparation for deployment, or treatment options if this were to become a disease of human consequence?
The WHO has prepared viruses as to make vaccines, so they are always updating the viruses that they have for vaccine production based on the most recent events. So that preparation is already happening and can be started to produce vaccines in a period of several months. So that is a possibility. With regards to treatment, there are several antiviral agents such as oseltamivir that can reduce the severity of disease and can be used. So those are basically the options that we have in case the virus jumps over to humans and starts spreading.
So at the moment, we would be reliant on the existing flu vaccine cycle and drawing on these potential strains, but using the same methods.
It could be the same methods, or it could be alternative methods, such as mRNA vaccines that could be used. Yeah, there are various possibilities, and certainly following the COVID-19 pandemic, there's a lot of lessons have been learned about how to rapidly produce vaccines.
And just lastly on that front, and there has been questions around the safety of milk, whether pasteurization is a fail-safe measure of securing milk supplies, and what's the US doing at this point?
In the United States, there have been studies of store-bought milk for the presence of the virus, and by PCR, I think about 25% of the milk samples tested positive for HPAI H5N1. However, subsequent virus culture showed that there wasn't any infectious virus present in those milk samples that were pasteurized milk samples. So pasteurization until now seems to be safe, make the milk safe for consumption. One caveat is that the requirements for pasteurization are to reduce the level of infectious virus by 10,000 so 10 to the 4. And if there are milk, if there is milk in which the virus has an even higher titer, so from individual cows, we know that there are even higher titers possible, up till 10 to the 10th, perhaps, then pasteurization would not be sufficient. So you can't rely 100% on pasteurization in a very strongly positive milk sample. But in general, it should be safe. This is different for raw milk. So if there is a HPAI virus circulating in dairy cattle, it would be strongly recommended not to drink raw milk, because raw milk with virus in it can bypass the acid barrier of the stomach quite easily. And we know from animal experiments, and we know from a few human cases where raw blood soup was drunk back in the beginning of the outbreak, that can lead to infection. And as you've seen, infection can lead to about 50% mortality. So that's something that really should not be done, to drink raw milk in an area where the virus is present in the dairy cow population.
Yes, we have to wrap up. I can continue with many questions, but just one final one around the oseltamivir use. The recollection is that during the Hong Kong outbreak, there was very rapid acquisition of resistance. Is that your knowledge as well? And is there any expectation that oseltamivir will provide durable treatment option? The history seems to suggest that it's not something to rely on.
Yeah, I agree that it would not be something to rely on. There are, however, several other versions of such antiviral agents that also can be used, that have a different mechanism, so a different resistance pattern.
So thank you very much. It's 5 o'clock, and you can see that everyone has stayed on till the very end. Just for those of you who have asked, it has been recorded and will be made available through the Sydney ID website with Thijs's permission in the coming days, but we will inform attendees when this is available if you want to share it with anyone else. It's been very timely, Thijs, highly informative, and a lot of food for thought. Thank you for sharing your expertise with us today.
You're very welcome.